your laboratory recently made a breakthrough, and that involves, I believe, stem cell delivery, making that much easier than before. So what kind of implications might this have in the way of new treatments to different diseases?
The power of stem cell, a lot of it really comes from its differentiation capability, transforming from the original type of cell to other cell types. And a major way of guiding the differentiation pathway is through sending in genes, sending in DNAs into the cell nucleus. And this method heavily depends on delivery technology. This is because stem cell treats the genes, the external genes, as foreign invaders. Stem cell have a set of barriers to just block the entry, all the way from cell membrane to intracellular vesicle barrier to nucleus membrane barrier, and things like that. So, and on top of that, stem cells are very well known to the biology community to be a so-called hard to transfect cells. So stem cells, immune cells, neurons are much more difficult to be transfected, that is basically sending genes into the cell nucleus, than pretty much all the other types of cells. And people don't really know why. And people also lack effective methods to deal with this kind of hard to transfect cells. And in this year's NanoLabor paper, we basically developed a technology to focus on addressing these bottleneck step. After overcoming vesicle escape in mesenchymal stem cells, we are able to improve the delivery efficiency by several folds compared with the conventional transfection reagents. We actually did a head-to-head comparison with the commercial transfection reagent, the gold standard reagent called lipto factor ME, and we found that we can give up to 6-fold improvement.
Hello and welcome to the Moral Medicine channel. My name is Eric, and today we're joined by a very special guest, and that is Dr. Ruan Gang, who is the co-founder of Core Quantum Technologies and was the research scientist at Ohio State University in 2007, before becoming a professor at the College of Engineering and Applied Sciences in Nanjing University in 2013. He then went on to become the senior associate professor at the Academy of Pharmacy at Xi'an Jiaotong-Liverpool University. And his laboratory develops nanomaterials-enabled technology for biomedicine. He has published 50 scholarly papers in peer-reviewed journals, and he has 10 patent applications, including one granted US patent and 2 patents granted in China. So thank you so much for joining us today, Dr. Ruan Gang.
It's great to be here.
And you have recently made a very interesting breakthrough in the realm of stem cell regenerative medicine. So I really look forward to talking to you about that today. But before we get into that, just for the audience, people who may not be too familiar with this field of medicine, could you briefly explain what the field of regenerative medicine involves, essentially?
Okay. Regenerative medicine is really a new way of treating diseases. So traditionally, diseases are treated either mechanically, that is basically surgery, using mechanical tools, treating people like automobiles. Another traditional way to treat patients is using chemicals, typically small molecules. This is basically treating our body as a chemical plant. So when some chemical reaction, chemical pathway goes wrong, you put in some chemical to fix the reaction. So regenerative medicine does not use mechanical tools or chemicals. It uses cells. So it is really a cell-based therapy. So the rationale behind this is that the fundamental unit of life is really a cell. Especially with this kind of cells called stem cells, which have regenerative power, has differentiation power to go from one type of cell to another. This is especially powerful. For example, the kind of stem cell we currently deal with in our lab is called mesenchymal stem cells. We can use them to differentiate, to transform to cell types like bone cells, heart cells, and even neurons. This can address a lot of the limitations of the conventional therapies based on surgery or chemicals.
Fascinating. Thank you so much for that explanation. Now, your laboratory recently made a breakthrough, and that involves, I believe, stem cell delivery, making that much easier than before. So what kind of implications might this have in the way of new treatments to different diseases?
Okay. As I just mentioned, the power of stem cell, a lot of it really comes from its differentiation capability, transforming from the original type of cell to other cell types. And a major way of guiding the differentiation pathway is through sending in genes, sending in DNAs into the cell nucleus. And this method heavily depends on delivery technology. This is because stem cell treats the genes, the external genes, as foreign invaders. Stem cell have a set of barriers to just block the entry, all the way from cell membrane to intracellular vesicle barrier to nucleus membrane barrier, and things like that. So, and on top of that, stem cells are very well known to the biology community to be a so-called hard-to-transfect cells. So stem cells, immune cells, neurons are much more difficult to be transfected, that is basically sending genes into the cell nucleus, than pretty much all the other types of cells. And people don't really know why. And people also lack effective methods to deal with this kind of hard-to-transfect cells. So what we did in the recent papers, we actually published a paper last year in ACS Nano, and this year we published another paper in Nano Letter. These two are basically a series of papers. The last year's paper essentially performed the first systematic quantitative study of how nanoparticles transport in mesenchymal stem cells and compare that transport behavior with a more common type of cells called HeLa cells. And by that study, we identified a potentially bottleneck step, the most challenging step in the delivery, that is the so-called vesicle escape. And in this year's NanoLabor paper, we basically developed a technology to focus on addressing this bottleneck step. After overcoming vesicle escape in mesenchymal stem cells, we are able to improve the delivery efficiency by several folds compared with the conventional transfection reagents. We actually did a head-to-head comparison with the commercial transfection reagent, the gold standard reagent called lipto factor Me, and we found that we can give up to 6-fold improvement in transfection efficiency.
Fascinating. So in addressing this bottleneck, so to speak, your team essentially found a way to bypass the stem cells' protective barrier to make it easier to deliver these particles into the stem cells by utilizing a polymer. So how did your team discover this exactly?
Well, we discovered this by a pretty silly method. Okay, it has a fancy name called screening.
Right.
So screening is a fancy name made up by the pharmaceutical industry. It literally means that you throw every possible method onto the wall and see which one sticks. So as I mentioned in the last year's paper, we identified vesicle trapping, vesicle escape as the bottleneck step. So after that, we essentially tried pretty much every method we can find, including some of the methods developed by our own lab, and see which one works. All these methods we tried in the past have worked well in the more common cell types, but in these stem cells, we found that most of these methods simply didn't work at all. And we found that we consider ourselves lucky. In the end, we actually found one that actually worked, and it worked really well. So we can only imagine what if we didn't find anyone? Then basically we can only tell ourselves, okay, we have tried.
Right. One thing that really stands out to me about this new method that your lab has developed is the fact that I think the implication is that scientists will be able to grow stem cells outside the body and then insert this specific genetic information into them, which then causes them to differentiate into a specific type of cell. And then once the cells have transformed into the target cell type, they can then be put into the damaged area of the body to help restore it. So could you maybe elaborate a little bit on this, Dr. Gang, and talk about some of the implications that this method will have for certain different diseases?
Okay. Yeah. So let me just give a more specific example, clinical example, that is heart failure. So you see, we just went through the COVID-19 pandemic. One clinical condition of COVID patient is really heart failure. Actually, this happens a lot in China. I actually know people who had this after after COVID. So this is the part one type of conditions, the so-called long COVID. Yeah, and currently there is no good drug that is really effective against heart failure. There were some drugs that can improve the symptoms of heart failure, but none of them can really cure heart failure. So whenever your physicians discover that you have heart failure, you pretty much just have to wait till it deteriorate, and you hope you die from other causes.
Right.
So regenerative medicine based on stem cells potentially can actually cure heart failure. This is because we are using stem cells to transform to healthy heart cells. So those cells are actually real healthy heart cells. And this transformation, this differentiation process, a lot of it relies on good, effective, efficient gene delivery technology. So in the with conventional gene delivery method to differentiate, to transform the stem cell to heart cells, the differentiation efficiency is extremely low. So you don't, in the end, you don't get a lot of product. So we consider heart cells as the product of this transformation process, right? So in the end, you don't get a lot of heart cells. So these heart cells are precious. Yeah, they are worthy of really human life. So what our technology can help is really to give people, give physicians, give doctors more of these healthy heart cells put into patient's body. So yeah. Oh, I guess I might add that if you can find a donor of healthy heart, so obviously you can cure heart failure. But yeah, heart donors are very hard to find. There is a very extremely long queue.
Right. Yeah, absolutely. And what are the next steps in terms of what your lab is working on in respect to this breakthrough that you found? What are the next things that you are targeting?
Right. So the next step, we are mostly interested in the application side. We are still working on some of the mechanistic understanding, that's for sure. But we are very interested in pushing these towards the clinic. So we are working with several physicians in different hospitals to really move these towards clinical applications. Heart is actually one type of cells, one area of the application we are interested in. We are also interested in some other applications as well.
Fascinating. And I believe that you also utilize rat stem cells specifically. Could you just elaborate a little bit on that aspect?
Okay, so one major reason why in the papers we use rat stem cells is really it's easier to get than human stem cells. Potentially in the clinic, when we actually use these on human patients, potentially human stem cells work better. But we actually don't know that. We actually are not completely sure. But for so far, when we just perform experiments on animals, we just use red stem cells, cells that we drew from rat's bone marrow. We also got some stem cells from other parts of rats as well.
Fascinating. And to ask you a little bit more of an abstract question, what are the things that you're most excited about over the coming years in respect to the field of regenerative medicine?
Okay, this is quite abstract.
Right.
Quite big. Yeah. Okay, let me try to answer this abstract question in a very abstract way. I think there are two directions I think I'm really look forward to, and our lab is also working on these two directions. One is really the clinical translation. So currently, there was some successful clinical application of stem cells. However, the success is still quite limited compared with the potential.
Right.
Yeah, so when the full potential is realized, I really see this as powerful as AI to our society, and potentially even more powerful because without health, we cannot enjoy a lot of the.
Very true.
Yeah. So application, clinical translation is one direction. Another direction is really adapting the cutting edge advances of life science research to figure out what's behind the regenerative medicine, how the stem cells multiply, how they differentiate, things like that. So currently, life sciences, the fundamental research part, how life works, is really advancing at a rapid pace. A lot of new methods are being invented. For example, omics method, multi-omics method. So currently, biology has entered a so-called systems biology era. Yeah, and I might add AI, artificial intelligence, can be quite helpful for figuring out this complex network going on in cells and tissues. So yeah, this is obviously not a comprehensive answer, but I hope this can help the audience to understand how what's going on in the field.
Absolutely. And I think the last question I'd like to ask you is in respect to the application aspect of this. Are there some parts of the body or certain organs where it's potentially easier to utilize this breakthrough of yours? I mean, you referenced obviously heart failure. The heart is obviously a very important and complicated organ, but are there other parts of the body specifically that you have in mind where, the clinic will be able to utilize these breakthroughs?
Skin is relatively easier. I say relatively to produce regenerative medicine or tissue engineering based product to be translated to the clinic. The heart is actually quite challenging, and the most challenging is probably
the brain.
Yeah, that's most challenging. But skin, perhaps bones, probably
Fascinating. Well, thank you so much for joining us today, Dr. Gang Ruan. It's been very interesting talking to you, and we'll surely be following very closely the progress that you and your laboratory make in this very interesting and novel field of medicine. So,
Yeah, thank you very much. I really appreciate you guys' efforts in helping the general public to understand the real process, the inner working of science. A lot of times, the general public only sees the end result,
right.
but they don't necessarily understand so much about the process. So it's the process is quite messy, actually. Yeah, I hope you guys can help the public to understand how the sausage is made,
Right. Right.
so to speak.
Absolutely.
Yeah.
Yeah. Well, thank you so much for coming on to explain in more detail, what you're working on and the implications of your work. So it's been very interesting.
Okay, thank you.
Thank you so much.